Today, I'd like to welcome you to this webinar. It's titled Cosmos Automated In Silico Tools for In Vitro to In Vivo Extrapolation. First, I want to let you know a couple of business items. The webinar is recorded, and a link will be sent to ASCCT members after the presentation is completed. Also, the lines are muted, as I said. After the presentation, you'll be able to ask questions, and if you could just put those in the Q&A, I will summarize and ask them to our presenter. And so I'd also like to call your attention to the ASCCT itself. We host a number of webinars on topics like this every couple of months. And we're an American society dedicated to providing an opportunity for scientists working in vitro and in silico fields to collaborate. We have annual meetings, and our fourth one will be this fall, October 1st and 2nd in North Carolina. And so just save the date for that, and we'll be sending out more details shortly. So with that, I'll go ahead and introduce our presenter. Dr. Alicia Paini has worked at the European Commission Joint Research Center within the Cosmos project, developing systems biology models for the past 3 years. She holds a master's degree in food science and technology and in food safety, and a doctoral degree in toxicology awarded by Wageningen University in the Netherlands, which was performed at Nestle Research Center. So without further ado, I'm going to go ahead and make Alicia the presenter.
Can you see the screen?
Yep.
Okay, so I can start?
Go ahead and start. Thank you.
Okay, so thank you very much, Christy, for this introduction and to have organized this webinar where I will be able to present some of the achievement of the Cosmos project, which we have worked at the JRC in the last 3 years. I would like also to thank all the people attending. And today I will talk about the computational tools developed to be able to perform in vitro to in vivo extrapolation in an automated way. The structure of today's presentation will be divided in 3 parts. The first part is an overview of the Seurat 1 cluster project and introduction to the Cosmos project. I will then briefly mention the computational models developed and show how these models can be applied in an automated way to perform in vitro to in vivo extrapolation. So I would like to start by giving some background information on the current European legislation on cosmetic products, which will be familiar to many of you. It all started with a first ban on final cosmetic products reaching the market in 2004, which was then followed in 2009 by a partial ban on single cosmetic ingredients within the European Union. And this partial ban was done because it was difficult to find alternatives for most of the complex human effects endpoint, such as repeat dose toxicity, which will include skin sensitization, carcinogenicity, reproductive toxicity, and toxicokinetics. However, the full ban on animal testing for cosmetic ingredients came into force on the 11th of March 2013. Since the introduction of this regulation, a lot of research and efforts have been put into the development of in vitro and in silico alternatives, since it's also a key point not only for the EU legislation on cosmetic products, but also for other EU regulations such as REACH. An example of investments towards the development of such methodology is the Seurat 1 cluster project, which is a €5 million project co-founded 50-50 by the European Union FP7 funds and the Cosmetic Europe Industries. This is a long-term initiative started in 2011 and will end at the end of this year in 2015. And with the Seurat 1 is made up of 6 research projects dealing with in vitro assay, stem cells, biomarkers, bioreactors, and computational tools. The projects are ScreenTox, HemiBio, Detective, NoTox, Cosmos, and ToxBank. ToxBank is actually the Seurat 1 data warehouse where all collect outputs will be placed at the end of the Seurat 1 project. We also have a coordinating project coach, which provides direction and feedbacks to the other research projects. Cosmos is the project developing the computational methods within the Seurat 1 and is a project of 6.7 million euro, coordinated by John Moore University in Liverpool, and counts 15 partners over Europe and 2 partners in the US. It aims to develop computational models to be used to perform safety assessment and to predict the effects of repeated dose exposure to cosmetic-related chemicals. Cosmos, as you can read, stands for Cosmetics to Optimize Safety. And if you would like to have more information, you can visit the website where the link is on the top right corner. I would like now to introduce briefly the outputs already achieved within Cosmos. The first one is the Cosmos DB, so was developed by Altamira Molecular Networks and the University of Bradford. It is a new database which brings together access to chemical structure as well as an inventory for cosmetic ingredients and the related toxicological information of these chemicals. The first public release of the Cosmos database, version 1, was done in December 2013. It's open source, and you can easily search the database in an easy, intuitive way. I would like to stress out that if you would like to have more information on the Cosmos database, a webinar was already recorded in February 2014 and is available at the following link, as well as tutorials of the Cosmos database. A second output was the establishment of a threshold of toxicological concern for the cosmetic ingredients. We all know that the TTC concept is used widely in the risk assessment process to refer to the establishment of a level of exposure for chemicals below which there will be no appreciable risk to the human health. So the concept proposed that a low level of exposure will, with a negligible risk, can be identified for many chemicals. So we can use this approach and extend this current paradigm for the application of the TTC in cosmetic ingredients. So also for this output from the for the TTC, you can find information in the Cosmos website, and a webinar was also recorded in July 2013. Furthermore, we are working in developing in silico models such as QSAR, chemiotype for toxicity, as well as a PBK modeling and virtual cell based assay. When we integrate the PBK modeling with in vitro testing, we can allow to perform in vitro to in vivo extrapolation, and we will have a better prediction of the safety of the chemicals. Of course, because of shortage of time, I'll be focusing today mainly on the work carried out to describe the approach to in vitro to in vivo extrapolation. And however, Cosmos computational tools were developed in several areas, like absorption for skin permeability and gastrointestinal absorption, for biokinetics, so physiologically based kinetic models, the human bioaccumulation factor models, and virtual cell based model. For chemical space analysis, we have the principal component analysis, functional groups profiling, structural alerts, and models such as receptor binding and profiling category building. However, for today, we will just focus on the biokinetics. In order to achieve the in vitro to in vivo approach, we need to build so-called physiologically based kinetic models. I would like to make a point that I prefer the acronym PBK, which is a bit more general, but it's It's the same as saying pharmacokinetic, toxicokinetic, or biokinetic. So the term kinetic is, in this presentation, just a general term to describe all physiologically based biokinetics, toxicokinetics. So the PBK model is represented here by interconnected compartments, represented the body, and is described by mathematical equations that can facilitate the quantitative description of the absorption, distribution, the metabolism, and excretion of a chemical, taking into account physiological and physiochemical parameters. After, of course, exposure of the body to a certain chemical. This exposure can be either oral, inhalation, or dermal. Such models can help you, can provide to you time profile and dose response curves and results. We build these models following the paper by Riches et al., where she describes in a clear way and how you can build up this model. First, we defined the conceptual model based on the mode of action of the chemical and the reaction within the body. We then translate this conceptual model into a mathematical equation. And here I'm reporting as an example the equation for the liver, where we can see that a mass balance equation describes the uptake of the chemical in the liver compartment, taken up from the GI tract, and then the flush from the arterial to the venous blood, and finally also the metabolism, because for mainly most of the chemicals under study, the liver is the target organ for metabolism. After this, when all the mathematical equation describing your model are set up, you can actually go one step forward to find all the values for your parameters. So the parameters usually that are needed are so-called the ones that represent the body, so physiologically and anatomically, so like tissue volumes, blood flow rates. And then you have physicochemical properties of the chemical, like partition coefficient, and biochemical parameters for uptake constant and metabolic parameters. All this information can be either found in the literature, in the by doing experimentally in vitro, or by in silico predictions. Once we obtain all this information, we can put our model into a software package that can solve the equation. So it can be MATLAB, Berkeley Madonna, but for the present case of the Cosmos project, we have used R package because it's a freely available tool. After we can, let's say, validate or evaluate the model against in vivo data, which is usually a really important step in the conceptual building of the model. And finally, we can make prediction. These models can, let's say, be used to extrapolate from in vitro to in vivo data, to use for acute to chronic toxicity data, to extrapolate from high doses, which are usually used in animal studies, to lower doses, which are more relevant to human exposure, to look at the inter and intra-species evaluation, extrapolate from a single dose to a multiple dose, or also extrapolate from one route of exposure to another route of exposure. Here, I would like to summarize. You can see all the PBK models that have been developed at the JRC. We have built for 10 chemicals with 3 different routes of exposure: oral, dermal, and inhalation. I would like to say that within Cosmos, we are not the only partner developing such PBK models. There is also Inaris in Paris, which is building PBK models for acetaminophen, valproic acid, and isoeuronol, and is now currently working on a generic PBK model script that will be really useful at the end to build in an automated way. I would like to also briefly mention that a simple PBK model was used based on only 3 experimental properties: hepatic clearance, protein binding, and renal excretion, and can be used to calculate human bioconcentration factors for chemicals in order to rank them based on their potency. And this was illustrated in a paper published by Tonetier et al. From our team in JRC. So the second biokinetic model developed within JRC is the so-called Virtual Cell Based Assay. This model tries to mimic what's happening actually within the laboratory setting. So before, with physiologically based kinetic models, we are describing what happens to the chemical inside the body, the human body or a rat body. However, here we are trying to mimic what's happening in the laboratory settings by taking into account all the experimental setup. So the virtual cell-based assay has one purpose: to simulate the fate of a chemical inside the well, taking into consideration how this can be, let's say, within the restricted defined compartment of the well, for instance, a 96 well plate, and can calculate intracellular concentration leading to cell perturbation. The integrated model is made up of 4 main sub-models. So a fate model, a cell model, a cell growth model, and a toxic effect model. Some publications have already been written by Josema and are available on the web. I would like to briefly say for each model a few words. So the fate model predicts the partitioning of the chemical in the well, so taking into account the consideration of the total concentration in time and the possibility to bind to the plastic, so the exchange to the plastic, the exchange or binding to lipids and protein, as well as well exchange between different wells, headspace evaporation, and a certain amount will be freely available and will enter the cell. In order to describe this process, you have mass balance equation as written here, and they are and you need parameters in order to input in order to run the models. And these parameters, such as the Henry constants, the air and water degradation, the molar volume, and atomic diffusion, can be achieved by Q-user prediction, experimentally, or of course via the literature. In our case, for all the models that we have developed, the VCBA, we have used APSuite to calculate these properties. Okay, here was the formula. So the virtual cell-based, after entering the cell, in the virtual cell-based assay, the chemical is uptaken in the cell, and the cell model is described by 3-compartment model, taking into account an aqueous, protein, and lipid compartment. And in the cell model, we also have metabolism, taking into account if, of course, the cells are metabolically competent. The descriptors needed for the cell model are the mass fraction of each compartment, so the protein, lipids, and water, and it can be found in the literature, partition coefficient within the cell, and the uptake rate. Then we have the so-called cell growth model that takes into account the cell population over time, and it's described by the Leslie matrix, which you can see here below in the slide. And it says that there is the probability of surviving and staying in one of the stages, the probability, which is translated to the P, or the G, which says the probability of surviving and growing into the next stage, as well as a fecundity rate. All this information, in order to run this model, you need information on the cell dynamics of the cell cycle. And if the cell doesn't proliferate, the model will be restricted to only remain in the G phase, so no actual cell cycle will be simulated. So for instance, for FRGs, the cell growth model is, let's say, set to 0 to 1 stage. So the final part is represented by the linking of the external concentration with the internal concentration and the effect experienced by the cell. So it can be a harmful effect like cell mortality, or also an effect like mitochondrial membrane potential. And in order to achieve this, we can run the model based on the two parameters, such as the no effect concentration and the killing rate. This can be simulated, but at the same time, you can use an experimental curve achieved in vitro in order to optimize the method and minimize the error of your model prediction. I would like to say that all the model equations were written first in MATLAB and then were translated into the R language. So we have both scripts available. In this slide, I would like just to briefly show how the output of the models you can achieve graphically results and are displayed. So on the top, you can see the constant, on the top left, you can see the concentration versus viability. On the top right, you can see the time profile curves versus the relative number of cells. So this represents the cell growth. And the concentration in time in the medium and the concentration in time versus intracellular. There is also availability to display the headspace and the binding to plastic. So the applicability of this model, it can be used by scientists that would like to refine their in vitro study and by a risk assessor to obtain an intracellular concentration leading to an effect, and can help this to actually define a new point of departure for the risk assessment project. For this type of modeling, we have here a list of the 25 chemicals and the different cell line for which we built the virtual cell-based assay. Simulation can be run in a single or repeated exposure. However, for the repeated exposure, we were able only to carry out experiments in-house with HepRGs and only for a few chemicals, so caffeine, amiodarone, estragol, and acetaminophen. But of course, we can always implement and use other available data in the literature, or if they're available there. In the single exposure, we have 4 different types of cell lines going to cardiomyocytes, HepG2, HepRG representing the liver, and A549 representing the lungs. And we would like also to extend the model to the neuro cells and to the kidney, if possible. I would like to briefly say that all the methodology behind these models are also available via the database service on alternative methods of EURL ECVAM. So from our web portal, you can see here the link. And if you would like to have more information, you can contact the scientist which is involved or visit just the web page that is given here. I would like now to just come together and combine the PBK model and the VCBA model in order to create a model to perform in vitro to in vivo extrapolation. So how, behind the scene, how this was done. So we first built a PBK model that was able to convert, as I mentioned before, from an external exposure dose to a concentration of the chemical in the organ, for instance, reaching the liver. Then this was integrated by integrating, into the PBK model a compartment representing the cell, as done, as mentioned before, from the virtual cell-based assay. The concentration of the liver, of the chemical in the liver, was translated to a concentration in the cell. And as an example of such an approach was reported by Gajewska et al. At the beginning of this year, or at the end of last year, that we see here in this table that she represented the dose response curve versus the concentration inside the liver and the intracellular concentration. So then in parallel, we can perform our in vitro experiments to measure the effect, such as loss of cell viability or loss of mitochondrial membrane potential or any endpoints that can be measured and can be translated into a mathematical equation. And this external concentration can be translated into an intracellular concentration. And by achieving the optimized NEC and killing rate, we can define an in vitro cellular concentration response curve. We can then compare the intracellular concentration and actually relate to the in vivo external dose to the effect by doing it in an automated way, and you can achieve this type of curve that you can see at the bottom right, where you have dose response curve versus your effect. So the full dose response curve can be used to predict, let's call, and used in benchmark dosing, and to set a point of departure for the risk assessment. And if you are, let's say, if you already have a dose that you would like to make a direct extrapolation, you can directly extrapolate based on the curve to the corresponding effect, or by, let's say, reverse dosimetry. So this is forward dosimetry, or by reverse dosimetry, going from the dose which gives, from the effect to the dose which gives you this effect. I would like to show that this approach was established for the following compounds of caffeine, coumarin, estragol, ethanol, and nicotine, because for this we had both the PBK model and the virtual cell-based assay. Unfortunately, for now, it's only possible for liver cells, but it would be nice to investigate also further for cardiomyocytes or other type of organs. Okay. So the here I would like just to briefly say I'm entering the last part of the presentation, and I just want to say that if you have a new, let's say, cosmetic ingredient, and you are in need to perform such extrapolation and to be able to predict in an automated way your effect, you can use our models, but of course this model can be a little bit tricky, so we try to make them in an easy way, and we are trying to develop automated tools for automated tools. So with the using the KNIME technology, which allow us to integrate all the models previously described in workflows. And here we have an example of the so-called physiologically based kinetic model workflow. We have a similar so these automated tools run on the KNIME platform, and they are so and we have also the virtual cell-based model. Also, the integration of the virtual cell-based model and the PBK model allow us to make a virtual workflow for virtual cell-based for the in vitro to in vivo extrapolation. And here we can see that for each workflow, we have an input zone where you can enter the parameters needed to run the model. And this is described by the green circle on the top. And then we have a model that also, a core zone where the model scripts are hidden. And finally, we have an output zone where we have the graphical representation that can be either displayed as a table or as a graphical representation. Furthermore, for the in vitro to in vivo extrapolation approach, in the workflow, we try to make an automated extraction of your input dose versus your predicted viability, or from your input viability to your predicted dose, in order to be in an easy way to extrapolate the output that you would like to achieve. So here you can see in the table below that there is an input dose of 100 milligram, which has a corresponding predicted viability extrapolated from the curve simulated by the tool of 0.4, 0.5. All the in silico model developed will be made publicly available through the Cosmos KNIME web portal, and also from the Cosmos space. And you can either download them to your desktop KNIME, so after installing the KNIME, which is an open analytical platform which you can use and we have used in order to translate the in silico models to ready to use and hopefully also easy to use automated tools. After this download of the software, you can download the workflow and use it, and you can have access to all the nodes which are behind the workflow. And you can also access the script, the R script of the models. However, if you don't need and you're not interested in to see the background and the R script or the mathematical equation, you can just use the online web portal version. For the online web version, you can register for free to this Cosmos Space and log in into the KNIME web portal by using the same credentials. And you can download guidance through the Cosmos Space, and you can find the links in this slide. So once you have registered and you access the KNIME web portal, you will find a public folder where you can click and you can see a subfolder for each of the areas that I mentioned previously, where we are developing in silico tools and where these tools are stored. So if you click on the biokinetic folders, you will find the PBK model and the virtual cell-based model, as well as the in vitro to in vivo extrapolation and the bioaccumulation model. Once you select the PBK model, for instance, you can reach the introductory page, which gives you a briefly overview of what the tool can do. And by clicking Start, you will access the page where you can input your parameters and you can set up your run. And you can ask the model to run by clicking Start. You can also select if there is one or more let's say script for that specific chemical. For instance, for caffeine, here you can see that we have 7 scripts for dermal, oral, for human or rat. So you can just select the script that you would like to use. For understanding what is behind this script, you should download the documentation that goes along with the workflow, which is available on the Cosmos Space. By clicking Run, after approximately 5 minutes, you will have results displayed as in this slide, where you can see that there is a prediction of caffeine and the metabolites in the blood, but as well as in the lungs, as in the liver. So for space, I just showed one graphical output, but it's nice because you can also have information on, displayed as a table with Cmax and AUC for the liver, the lungs, and the blood, and for each the parent compound and the metabolites. And this can be downloaded, the report can be downloaded as a PDF or Word or in other formats. We also made available as a tool the bioaccumulation model. For this bioaccumulation, we have developed 2 workflows, one mimicking what's reported in Tonnelier et al., and a more general one where we can, where you can input your chemicals and properties and all the information that you need if you have a new chemical that you want to run and to use for this simulation. Here you can see an example of the output of the bioaccumulation tool after a run. It takes also around 5 minutes, so it's quite fast. A similar tool was developed also for this VCBA, so for the Virtual Cell Based Assay. And here you have, again, an introductory page explaining a bit what the model can do and also what type of information you need in order to run the model. For the VCBA, of course, you need more input parameters, or you need to select at least the cell type, the chemical that you would like to test, if you would like to test in single or repeated exposure. You can select the number of cells that were in your experimental setup. Look at the simulation of the, takes into account the exposure of the cells, so the time, total time, for instance, for 24 hours incubation time. The assay volume, which is in cubic meter, the initial concentration, and the last 3 are actually for the repeated exposure, so the initial concentration of the repeated exposure, and then the interval times for that you have selected your let's say make the repeated exposure, and the number of exposure that you did with your plate. So for instance, if you have a 1 week experiment and every 24 hours you will refresh the medium, you set up this information into the virtual cell-based assay. And finally, you should also enter the concentration range used in your experimental setup. You then run the model. Unfortunately, it takes around 20 minutes for the model to run, so it's still a bit slow. And at the end, you will achieve curves like I showed in the previous slide. And here are reported, for instance, for the concentration versus the viability. I would like also to mention that for now, it's limited to the chemicals that we have tested and for the cell lines that we had information on. So, of course, if there are available other data, we can include them inside the model. We also have introduced the in vitro to in vivo extrapolation. So you can select here the initial dose, the interval time, the number of doses, and select which type of model you want to run. You then can use the last two boxes are actually for this extrapolation, direct extrapolation from the curve that the tool will simulate. And you then can display your results in a table, and this table can be downloaded in the report, and you can use it for further analysis downstream in your process. So here is actually the results. As I mentioned, you can see how the report looks like, in the meaning once you can download, after downloaded, you will have this table with your input dose and your input viability and predicted dose and predicted viability. So so for all the in silico tools that we have developed, both in the biokinetic area but also in the other areas, there are written guidelines, and also tutorials will be made soon. They're under construction now, but they will also be made available, and will be you can access them via the Cosmos space. So I would like to conclude by saying that I think we have developed a new type of models that we can use as alternatives to animal testing, and can be used for regulatory purposes. And I would like to ask who is interested to please test these models from the Cosmos space or the KNIME web portal, and please send us feedbacks, because there always need to improvements. We always find some bugs and mistakes. So I would just like to ask you to also like be kind of part of this project in order to test them. I would like to mention that this is the third of webinar organized between Cosmos and the ASCCT, and there will be more to follow. And if you have any questions or information, you would like more information about the Cosmos project, please visit our website. And if you don't find the information you're looking for, please email our contact address, and we will be happy to provide any information. For questions of the webinar, of this webinar and this presentation that I made, you can ask information to me or to our common JRC email address. And also, if you have feedbacks on the models that I presented today or more questions, please feel free to email to my email address. I finally would like to say that this was a work team effort. EURL ECVAM scientists have been involved. It was all initiated 4 years ago by Jose Zaldivar, and all the PBK models were mainly developed by Monica Gajewska, as well as with the help of Jose Salabenito, which also helped in the translation to the KNIME of all these models. We had toxicokinetic advice from Jos Bessem and guidance from Andrew Worth. And I would also like to thank Alexander Perry for advice on the PBK model and following Monica's work, and Thurston for helping with the KNIME implementation, as well as Andrea for reaches for guidance and constructive criticism on all the outputs that we are generating. And finally, I would like to thank all the Cosmos partners, and I will now leave the floor for questions. Thank you.
Okay, well, thank you very much, Dr. Paini. Let me get right to the questions. We have, the first question is from someone from RIFM, which is the Research Institute for Fragrance Materials, not RIVM. But the question is about the virtual cell and the IVIVE model, and they talk about, they say that many people use a tenfold margin when you're extrapolating from individual to population, from animal to human, a sort of a 10x safety extrapolation. You might be aware of that. And sometimes people argue for a threefold factor. And the question is basically, I think, do you think that these models could be used to, I guess, provide a little more of a science-based margin than a 10x or something?
Yes, I think this is actually what we are trying to do, is to provide a better estimation of safety assessment factors in order to fill in gaps that are these safety factors taking into account. So by use of this in silico models, so the VCBA and PBK modeling, and also the in vitro to in vivo approach, I think we are going one step further in more biological description and taking into account more the toxicokinetic and toxicodynamics of the human. And we are developing more tools that are human-based and not anymore. There is not anymore so need to extrapolate from animal assays, let's say. So I think, yes, this is the way forward, and I think, I hope that one day everything will be, let's say, translated into mathematical equations so that we will be able to perform a better risk assessment.
Right. Another question is very similar to one that I think I had. I was intrigued when you said that for the virtual cell-based assay, you're looking for more data to sort of help refine the models. And one question is, what data do you need to do this exercise for a different goal?
So the data that I need, so I try to, in the theoretical part, when I describe the models, I try to say the parameters that are needed. And so the most important, which for the fate model are descriptors characterizing the, say, like the chemical fate in the model, so the partitioning and these things. And this, for the VCBA, can be done by using in silico tools like QSAR. So this is not such an, this is not difficult to achieve. However, for instance, for describing a cell, we need information on the cell type. We need information on how the cell behaves inside the in vitro system. So for instance, if you have a cardiomyocyte, the shape of the cardiomyocyte will be different than a spheric shape, how we now represent the hepatocytes. If you have a metabolically competent cells, we need to know all this information. We need to know if they proliferate, the information on the cell cycle and how this, all this information. And finally, we would need some in vitro testing on the, let's say, toxic effects or the effects that one would like to measure. So for now, the model is based on cell viability and mitochondrial membrane potential. So if there are information type of concentration curve. So for optimizing the model, we would need curves describing, taking into account the dose range, so the concentration range, sorry, and the effect of this simulation. So this would be the most important, and the type of cell. So if somebody would like to provide us for a certain chemical a concentration response curve, we can try to input this chemical inside the VCBA. And if it's one of the 4 cell lines that I already mentioned, we already have all the information. But if it's a new cell line, we will need information also for this cell line. So there is a lot of things that we can do and can be done.
Great. Another question is, specifically about protein binding constant in order to determine the in vitro fate. I think you mentioned protein binding, but they're saying this is a difficult parameter to find.
Yes. Protein binding and plastic binding are really difficult parameters. We have, luckily, that's why our data set is also restricted, because they were performed in-house a couple of years ago, and we have always been using the same data. So, of course, also this could be refined, could be retested, and could be, re-evaluated, let's say, also for the model, because of course in 3 years that we are now working on it, it can be that we think that maybe the methodology we have used for protein binding at the time was different, or we could use a new way. And if there is this possibility, of course I'm not an expert in the area of the protein binding assays, but if there will be the possibility to perform, of course we will be happy to have information.
Great. Another question asks, can you please comment on the applicability domain, in other words, the chemical space covered, of the IVE model? And I think you did mention the chemicals.
So you mean how many, you think it's how many chemicals we have included, or we have? So of course, all these models, I would like to say, the best would be if you would have a generic tool where people can input their chemical and run the models, and with the inputting input parameters and the values achieved by their research, and then they can use the tool to extrapolate. But unfortunately, we are not at that stage. We are at the stage that we are still developing for chemical specific. So for now, we have done for 5 chemicals. So for 2, for caffeine, coumarin, they have been established, and for caffeine, it's already available online. We are now working on estragol, and after there will be ethanol and maybe also nicotine that will become available. And the restriction is that we will need information on the PBK model, so that there should be PBK model plus information from the virtual cell based assay. So that's why it's a bit restricted at the present moment.
But just to emphasize, if I'm correct, there is a general model for the first model.
So it's really difficult to really have a generic model when you are looking at for instance, for the physiologically based kinetic models, because for chemical specific, you know the mode of action, and you can let's say implement based on the mode of action your PBK model. So if in the liver it's metabolized into 4 metabolites, you can take count of the metabolism. For the bioaccumulation model, which was a simplified PBK model, which takes into account renal excretion, hepatic clearance, so the metabolism, but in a really simple manner, so yes or no, and also protein binding, then there we were able to actually set up a more generic model where you enter your chemical name, you enter the parameters that you need to run the simulation. And if you look into Tonnelier et al., you will understand how the model was built. Today I was not able to go too much into detail on this specific PBK model, but I think this is a way forward in order to translate this model into more generic models and we wanted to show how we could apply what would be actually the last output from the project to have these generic PBK models. So, and also the generic VCBA model. So that would be the ultimate output, let's say, at the end of the year, hopefully.
Great. So another general question, what do regulatory authorities think about these models?
I have. So I'm a bit, I think that I'm not really into contact with the regulatory people. I think nowadays by having discussion also with EFSA people, they are more and more looking into such in silico computational tools. However, they are still a bit in their infancy, and they are not really keen to let's say use and go away from the old-fashioned risk assessment way of dealing with risk assessment. So it's a bit difficult to really make them. I know that they are aware, I think, of all the work that have been done, but yeah, there's still a little bit of difficulty in to make them accept this type of models, I would say. But yeah.
Okay. One question about how have the models been validated, and how reliable are the output values?
Yes. So this is a really good point, an important point, because all models have a lot of assumptions, and of course they would need to be validated. The PBK models are validated especially on certain persons, so they were all validated, the PBK models, using in vivo human data. So that's why we also have restricted amount of chemicals, because for these chemicals we were able to find in the literature in vivo data. For the virtual cell-based model, this is a good question. It's still ongoing. How the best to evaluate and make kind of let's say validation of this type of tool, because it's for now what we are doing is just trying to optimize versus the in vitro concentrations, those response curve achieved in the laboratory, and then you get optimized simulation, but it's of course based on that type of cell line at that moment. So it's still yeah, a drawback. It's not really there. Yeah, it's really important to validate. That's it. Yeah.
Okay. We have one more question, unless someone else is a quick typer, which is, could you please comment on how the virtual cell-based assay method can help with risk assessment for sensitive subpopulations? And they say, for example, children, but I wonder with the cell types you have, whether disease status might be a better question there.
No, sorry, could you just reformulate?
Yeah, so basically, are there characteristics of sensitive populations?
No, unfortunately, in the virtual cell-based assay, we are only looking at representing the what's happening in the laboratory, so in the cell, and try to use this information in order to link them with the PBK model to a in vivo effect. So we should actually go for the sensitive subpopulation. We should go and look at the PBK model. So within the PBK model, then you can set up a PBK model, especially for children. At the present moment, we have established and built only taking into account adults which are healthy, let's say. But of course, this PBK model, once they are developed, and if we have a generic model, you can then set up, let's say, information just concerning the children. And I'm not sure if you have a certain type of disease, if, of course, this then becomes a little bit too detailed, but I think it can be taken into account. But then we will need a whole new project just to set up all this information for PBK modeling.
Yeah, I think you're right. I think it's possible to develop tools like this, taking that into account, but this tool in particular was developed for
in vitro
the
testing,
viral
yes.
cell, yeah. Okay, well, we don't have any more questions, so thank you again for your presentation, and thank you all for listening and participating. Just a reminder that the slides will be available on the Cosmos website, and a link will be sent out once those are all put up there. And as Dr. Paini said, I would encourage you to visit the Cosmos space and play around with the tools and provide feedback. And finally, I would also encourage you who are not members of the ASCCT to visit our website, which is ascctalks.org, to learn more about our activities and future events. Thank you very much and have a great evening or day.
Mm-hmm.